Hey everyone, in this video, I'm going to talk about the difference between DNA and RNA as part of our review for Unit 5, information that we've learned up to this point, or maybe even in 9th grade, to help us moving forward on this topic. So in my T-chart, the first thing I want to talk about is the difference in the structure between DNA and RNA. So DNA is double-stranded while RNA is single-stranded. And this is because of the structure of the nucleotide, which we'll talk about later. Next, we're going to talk about the size difference between DNA and RNA. DNA is much larger than RNA, usually containing millions of nucleotides, whereas RNA will probably contain hundreds to possibly thousands only. Next, I'm going to compare the location of DNA to RNA, and this is most contrasted or seen in eukaryotic cells because of membrane-bound organelles. So keep in mind that prokaryotes do have DNA as their genetic information, and it's found in the nucleoid, which is just in the cytoplasm. With eukaryotic cells, there are actual membrane-bound organelles. So for this comparison, I want to focus on eukaryotic cells. And in the case of eukaryotic cells, the location of DNA is going to be primarily what everyone thinks of, which is the nucleus. But remember that there's also DNA found in the mitochondria and the chloroplast. RNA is going to be found primarily in the cytoplasm. At any given moment, it may be attached to a ribosome, but the key feature here is that functional RNA is going to be found outside of the nucleus. We're also going to compare DNA to RNA in terms of their nitrogenous bases. DNA will contain adenine, thymine, cytosine, and guanosine, whereas RNA will not contain the thymine, it will contain uracil instead. Next and last, I want to talk about the difference in the sugar. In DNA, the sugar is going to be deoxyribose, and RNA will contain ribose. So let's look at this RNA sugar a little closer. So here is a 5-carbon sugar. Remember that from your oxygen, we number the carbons going clockwise. And here at carbon number 2, you'll see that there's a hydroxyl group here. This hydroxyl group is what makes this ribose. Whereas when we look at deoxyribose, the sugar is going to look identical, except that notice here, I've deleted the hydroxyl group. So this is the deoxyribose and this is the ribose. When you want to tell the difference between the two, you look at carbon number 2 to check to see if there is a hydroxyl group present in each of these. Now, one thing I do want to point out in both of these is you'll notice that I drew a hydroxyl group at carbon number 3. This is going to be important when we're doing dehydration synthesis and creating polymers of nucleic acids, because this is where we're going to get some of that water. Remember that water is H2O. So we're going to get some of that water from this carbon number 3, no matter, or that hydroxyl group at carbon number 3, no matter whether we're making ribose and deoxyribose. But to be able to tell the difference between these two sugars, you need to look at carbon number 2 and check to see if oxygen's present. I hope this video was helpful. Thanks for watching. Bye.